Welcome to the Network Systems Analysis Bootcamp, sponsored by the FGTB Council of the American Heart Association. Prior to attending the classroom session of the bootcamp, please review the following presentation. These slides serve as a primer to understand basic principles involved in high-throughput data interpretation using network systems analysis, and they prepare you for topics that we will cover in the bootcamp exercises. Hopefully, these resources provide sufficient background to allow you to initiate systems analysis of your own high-throughput datasets. Current omics techniques now generate tremendous output. mRNA, microRNA, proteins, metabolites, with lists typically running into the hundreds or thousands of IDs from a single experiment. With a high-confidence list of IDs and corresponding differential expression measurements in hand, what methods should be applied to integrate data and extract accurate and comprehensive functional insight? A focus on limited subsets reduces complexity, but comes with major drawbacks. For instance, selecting a smaller number of only the most extreme changes simplifies interpretation, but immediately sacrifices much of the acquired information. Not only is this counterproductive to the use of high-throughput methodologies, but this strategy may not even reveal the most relevant or consistent functional changes. Unfortunately, confirmation bias is also quite common when confronted with high-complexity datasets by scanning specifically for familiar genes or proteins, ones that fit initial hypotheses, or that are consistent with past findings. In either scenario, mechanistically relevant data may inadvertently be marginalized or ignored when arbitrarily restricting the scope of data analysis. As omics datasets continue to increase in magnitude and complexity, robust analytical strategies are vital for inclusive data interpretation and analysis. Together with increased high-throughput data acquisition, concomitant advances have occurred in complementary technologies, including establishment and growth of data repositories, strategies to systematically document and catalog molecular information, and computational approaches by which to screen and interrogate acquired data using these resources. This presentation will focus on these inclusive and evolving systems methods with an emphasis on network analysis. Biological molecules function in intricate webs of interactions, so networks compiling these interconnections provide a rational means of assembling data in functional context. As an example, the minimal network required to incorporate a complete set of differentially expressed genes or proteins via established interactions yields an immediate focal point within an experimental system, regardless of extent of dataset size or breadth of constituent functional variability. Networks are amenable to compositional assessment based on function ontology overrepresentation and enrichment analysis, and to structural examination, such as architectural topology or positional relevance of constituent parts. This enables mechanistic interpretation and informed hypothesis generation from collected data. Network biology thus offers a logical, inclusive approach to organize and interpret complex systems behavior and a versatile platform with the capacity to incorporate high-throughput data of theoretically unlimited size. This presentation covers the following subjects, with much of the time allotted to aspects of the final topic. Initial data reduction steps, such as heat maps and principal component analysis, will be overviewed briefly. We'll also look at one example of a useful online tool designed to accomplish both. Next, we'll quickly cover ontological classification and the gene ontology, the structured vocabulary that has facilitated development of functional enrichment analysis, and a few popular tools for ontology analysis will be mentioned. Expanding on these approaches are pathway analysis platforms that broaden categorical assessment beyond gene ontology terms. An excellent resource outlining hundreds of pathway analysis applications is highlighted, along with a general idea of pathway analysis output data formats. The second half of this presentation will focus on the final topic listed here, networks, their compositional characteristics that impart additional emergent properties, and how networks can be utilized to enhance interpretation of high-throughput data. Heat maps provide a nice visual representation for how individual samples cluster together or apart. The color scale generally depicts z-score transformed data, normalized to standard deviation units from the median or mean for each molecular entry. This allows for comparable image intensity regardless of the dynamic range of absolute abundance across all measured entities. Heat maps thus provide an overview of the entirety of your data, or a limited subset as shown in the larger and smaller heat maps, respectively. There are many strategies for generating heat maps. Two of the more common approaches are k-means clustering, which segregates data into a user-specified number of groups in each dimension of the heat map, and agglomerative clustering, which arranges genes or proteins in pairwise fashion based on closeness of expression correlation. The heat map shown here is an agglomerative heat map. Statistical expression correlation can also be used to generate interaction networks, which will be touched on briefly later in the presentation. The lower heat maps all represent the same subset of IDs and expression values, with the neutral z-score entries changing from black to white between the first and second maps, and then the high z-score color is switched from yellow to orange between the second and third maps. These demonstrate different perspectives and contrasts that may be chosen to emphasize a particular aspect of the map. Of note, most early heat maps were colored red and green, which should be avoided, as this color combination cannot be distinguished by individuals with the most common type of color blindness. Another common data clustering method is principal component analysis, or PCA. This provides data reduction to the sample level and thus high-level distinction between groups as well as the individual samples within each group. As a result, PCA is also useful for detection of intra-group outliers at the initial data analysis stage. Loading plots, as shown at the bottom, can also be generated to indicate the relative contribution in each principal component for every entity, gene, protein, etc., that contributed to calculation of the sample-level PCA plot. Both heat maps and PCA can be inclusive of all data or focus only on a limited subset. While providing useful overviews that cluster entries by similarity measures, they do not indicate which entries are differentially expressed, and they give no sense of either the functional characteristics that segregate one sample or experimental group away from another, or the nature of any functional relationship to the data. Clust Vis is an example of a freely available online tool that runs a number of R packages for heat map and PCA clustering and visualization. It was built using Shiny, an R package for interactive web apps that incorporate a variety of R tools to create a user interface that doesn't require users to have a working knowledge of R. Clust Vis is thus an example of a useful advance for biologists who lack coding skills, yet still have the need to make use of the various tools developed in the R environment. Many pathway analysis applications also include data reduction tools such as these, and for those that are more comfortable in a programming environment, there are also many R tools and packages available for biostatistical data analysis. Although heat maps and PCA provide attractive visuals, they don't highlight biological process or molecular function similarity amongst clusters or that distinguish between clusters or experimental cohorts. One way to begin accomplishing this is by categorizing data by its biological composition. This is often conveyed in manuscripts as a pie chart or histogram of functions, processes, or cellular compartments. While somewhat useful, these simple allotments fail to convey the fact that many proteins, RNA species, and metabolites are not limited to a single function or process, but often perform multiple roles, occasionally in multiple locations in the cell, and may even have distinct roles isolated to different cellular compartments. To properly account for these combinatorial effects, a uniform approach to evaluate and assign categorical representation is required. Moreover, it must be built on current knowledge compiled in the literature, be adaptable for addition of new information in entries and concepts, and be broadly applicable across the many species represented in molecular databases. Data interpretation is thus facilitated by structured descriptions of accumulated biological information that are readily accessible for interrogation. Fortunately, in the late 1990s, the Gene Ontology Consortium had the foresight to begin formalizing the description of gene products in different databases. The Gene Ontology Project is a collaborative effort to address this need. It uses structured vocabularies, ontologies, to describe gene products on the basis of three characteristics: biological process, cellular component, and molecular function, and it organizes these in a species-independent way. The gene ontology effort aims to achieve this by developing and maintaining ontologies, annotating gene products, and developing tools facilitating creation, maintenance, and use of ontologies. These structured ontologies make it possible to interpret high-throughput data in a systematic manner. Using the assignment and overall representation frequency of ontological groups and categories, it's possible to define statistically whether particular functions or processes are overrepresented or enriched in the totality of subcategories of acquired data. For instance, you can determine whether a profiling method enriches for specific categories by comparing a complete list of IDs to its relevant background, such as the known transcriptome or proteome. More frequently, it's applied to compare differentially expressed IDs to either the entirety measured, such as all genes on a transcript array, or to its representation relative to all ontological assignments for the full organism, which would apply more to, say, current RNA-seq or proteomic studies. Finally, enrichment tests can be used to determine the probability of observed overlap between two distinct experimental groups or treatments. In other words, for treatments A and B, what is the probability that they have precisely X upregulated genes in common, given the specific upregulated genes identified in each? Careful selection of an appropriate background or reference is critical, as differences in background will influence and alter predicted enrichment outcomes. The two most common statistical tests for calculating enrichment and overrepresentation are the hypergeometric distribution, the equation of which is shown in the upper right here, and Fisher's exact test. The distinction between these two, if you're at all interested, is that the hypergeometric distribution is the equivalent of a one-sided Fisher's test. An important consideration for these probability calculations is that multiple tests are running simultaneously, which can number in the thousands based on the number of IDs in a dataset. As a result, it's important to include multiple test corrections to define statistically significant probabilities, usually by Benjamini-Hochberg false discovery rate or Bonferroni family-wise error rate. Some of the more popular gene ontology enrichment analysis tools include David, Gorilla, Panther, and Amigo. Output from these tools is generally in the form of histograms or tables ranked by significance of enrichment in gene ontology-specific terms, specifically biological process, molecular function, or cellular component. Other tools, such as gene set enrichment analysis and the newer Enricher, have a bit more versatility, as they will also define enrichment against previously published datasets or data subsets. Gene set enrichment analysis applies different statistical tests, but it is one of the earliest tools developed for enrichment analysis. There are also functional enrichment applications designed specifically for metabolomic data, such as EMBROL, Metabolites Biological Role, and MetaboAnalyst, which incorporates a metabolite set enrichment analysis that they indicate is similar to gene set enrichment analysis. Many pathway analysis tools incorporate enrichment analysis internally against standard gene ontology terms. Often, they also provide additional functionality in defining enrichment in areas such as protein domains, sites of expression, such as normal tissues, cancer tissues, cell types, and cell lines, metabolic and signaling pathways, transcription factor promoter regions, and even clinical synopsis phenotypic terms. Several of these are integrated in the Enricher analysis tool as well. For anyone unfamiliar with pathway analysis, a great place to start is PathGuide. This resource, hosted by the Bader Lab at the University of Toronto, now lists nearly 700 pathway resources as of September 2017. And these span an array of application categories, as noted here. PathGuide includes hyperlinks to each listed resource with details on whether they are freely accessible, free to academic labs only, or if they require a paid license. In addition, some resources specify whether uploaded data must be in a particular standardized format, such as BioPax, which is the Biological Pathway Exchange, SciMi, the Protein Standards Initiative, Molecular Interactions, or SBML, the Systems Biology Markup Language. Indeed, proper data formatting is an important consideration when using any bioinformatic tool. To run pathway analysis of a dataset, you upload either a simple list of IDs, or a detailed spreadsheet including a list of tool-readable identifiers, generally in a common database format, such as human genome organization gene names, or some other identifier, together with expression values such as fold change, log2 ratios, and p-values or associated false discovery rate values for any statistical tests. Again, it is possible to examine your entire list without applying cutoffs, or by restricting with fold change or p-value criteria, or both. One can interpret upregulated entries, downregulated entries, or combine both to look at all significant differences together. It's actually worthwhile looking at all three, as the combined data may reveal things not evident from either up or down cohorts in isolation. Other criteria can be applied to enforce more or less stringency on search parameters, including specificity for organism, cell or tissue type, disease or mutant consideration, experimentally observed or predicted interactions, and can include direct relationships only, or also add indirect relationships, where expression of A is known to affect B, but the relationship is either undefined or acts through intermediary C. In this regard, it is often worthwhile evaluating and comparing both narrow and broad search parameters. Pathway analysis output typically includes gene ontology-specific enrichment, as you would obtain with gene ontology tools, but depending on the software and its range of search parameters, a number of additional categories might also be represented. This can include processes, functions, and components unique to mutants or diseases, metabolic and signaling pathways, including pathway overlaid and colored by the presence of differentially expressed genes or proteins, upstream regulators or enrichment of transcription factor promoters that may be predicted by or enriched within the data. If differential expression directionality is included, it might also be possible to predict whether a transcription factor or other regulator is activated or inhibited. Keep in mind that these are predictions only, and predictions such as these should be validated by experimental follow-up. Finally, the more comprehensive pathway analysis tools can be used to show known or predicted interaction partners via physical or regulatory interactions as networks. These can include proteins, DNA, RNA, microRNAs, metabolites, or drugs as interacting components. As a result, these more comprehensive commercial pathway analysis tools appear in multiple resources noted in PathGuide. Pathway analysis applications will identify and prioritize metabolic and signaling pathways, molecular functions and processes, and disease or development associations that are generally categorized by software vendor curation of the literature, topics that are outside the scope of standard gene ontology tools. Data can be generated in similar formats to gene ontology tools, such as histograms, heat maps, and tables, but can also generate useful pathway and network diagrams that superimpose and arbitrarily color differentially expressed components within the respective images, as seen here. Pathway analysis is best used as an interpretation tool facilitating hypothesis generation for subsequent follow-up, as I mentioned before, rather than as a confirmatory or validation step within the data analysis pipeline. It provides direction to interpret high-throughput data, which is best addressed by validation through experimental follow-up. Networks can actually be quite useful to guide this subsequent follow-up. While pathway analysis can specify a comprehensive list of network interactions, it isn't ideal for network-oriented follow-up for three reasons. First, pathway analysis tools are somewhat limited in terms of the size capacity that they can handle, in other words, how many network components they can visualize. Second, they lack versatility in network layout and visualization. Third, they are not designed to evaluate complex network analysis parameters such as network topology and secondary structure. However, there are network-specific visualization and analysis tools better suited for this. One of the most versatile and useful network analysis tools is called Cytoscape, which we will get to shortly. Since we'll spend the rest of our time focusing on networks and their applicability for biological high-throughput data interpretation, it's first important to understand a few, but definitely not all, basic network definitions and principles. Let's start with the definition of a network. In the simplest sense, a network is a collection of binary interactions, either between two distinct entities or an auto-interaction of an entity with itself. In the context of high-throughput omics readouts, these are molecular in nature, genes, proteins, microRNAs, etc., which are defined as nodes. Interactions between nodes are defined as edges, and represent one or more interdependencies or relationships between the nodes. In biology, these may be physical, regulatory, or genetic interactions, and may be represented as undirected or directed, either unidirectional or bidirectional, as shown in the diagram. Edges can also include additional information regarding relationship confidence or function. Node degree is the number of connections that a network node has with other nodes, with the degree distribution being the frequency distribution of measured degrees of all nodes within a network. Node clustering coefficient is determined by the proportion of nodes directly connected to it, in other words, its nearest neighbors, that also connect to one another, providing an indication of neighborhood relatedness or the extent to which nodes within a network cluster together. Biological networks are postulated to exhibit power law distributions of node connectivity, which is defined as a scale-free structure, and hierarchical organization with clusters of highly connected, interrelated nodes as modules. This in turn influences emergent network properties. Finally, network biology is the analysis of molecular interaction networks through complex network theory, comprising non-trivial topological features characterizing symmetric or asymmetric relationships amongst discrete network objects and their known or established functions and interactions, which can be used to guide medical or biological interpretation. So, let's go into a bit more detail to help you understand networks. Here's a small 6-node network, shown same on the left and right, to convey degree and clustering coefficient. On the left, nodes labeled A, B, and I have degrees of 3, 1, and 5 respectively. Once these are determined for all nodes in a network, their distribution can be plotted as the proportion of nodes with a particular degree, or pk versus k. On the right side, we'll determine the clustering coefficient for node I. With a degree of 5, node I has 5 nearest neighbors, all the other nodes in this network. Its clustering coefficient is the proportion of connections present between its nearest neighbors, of which we see there are 2, relative to the total number of possible connections between those neighbors, which in this case is 10. This gives node I a clustering coefficient of 0.2. After calculating the clustering coefficient of all nodes in a network, we can plot this distribution as well. Although relatively simple parameters, degree and clustering coefficient distributions convey important information regarding overall network architecture. Historically, it was believed that networks were arranged in a random connectivity pattern, which would result in Gaussian arrangement for the degree distribution, centering on the mean degree of all nodes in the network, with clustering coefficients similar for all nodes. In the late 1990s, a number of investigators, Watts, Strogatz, Barabasi, Newman, and others, reported that networks were in fact not randomly arranged, but instead exhibited what approximates scale-free connectivity. In this case, most nodes have very few connections, and a much smaller number of nodes have extremely high connectivity, which on a log-log plot in the center approximates a straight line. Regardless of the scale of these networks, they exhibited this same degree distribution. While this did not necessarily translate to the clustering connectivity, it was also found that many networks, including biological networks, exhibited a hierarchical structure that results in highly connected network modules with scale-free clustering coefficient distributions as well. Most networks today are believed to harbor scale-free or hierarchical architectural properties, although the network field still hotly debates whether this structure is in fact scale-free or instead follows similar but distinct mathematical properties. To quickly round out a few additional structural attributes and terms, let's take a look at another example. In this particular artificial network, node 21, which is much more highly connected than any other node, is termed a network hub. The clusters circled in gray are modules exhibiting high clustering coefficients. In biology, such network modules are often formed by protein complexes. Node 26 has a much lower degree than node 21, but serves an important role as a bridging node between a high portion of other nodes within this network. If the total number of paths connecting every pair of nodes within this network were calculated, node 26 would lie on nearly as many total paths as node 21. Thus, despite its low degree and its absence from being a part of any module, it may still serve a critical role as an information conduit for a large proportion of the paths within the network. While all these attributes may seem rather esoteric from a biological standpoint, together they confer emergent properties relevant to any type of network, including those in biology. As an example, we'll look at robustness. So what does robustness have to do with biology, and what do I mean by robust? Well, in a scale-free or hierarchical network, because of the degree distribution where most nodes have low connectivity, the random loss of any one node in a network is far more likely to occur to a low-degree node than to a high-degree node. Thus, this should have minimal effect on the overall network integrity, and in turn, the function or functions pertinent to that network, indicating that network structure is robust. On the other hand, this also means that networks may have one or more Achilles heels, as targeted removal of a highly connected hub could have a critical impact on network functionality. Thus, it may be possible to use network structures to identify potential nodes for targeting to validate a network's predicted functional associations. We will actually look at a biological example of this in class. Networks may be generated and assembled from a variety of information using different assumptions and input data. First of all, they can be assembled by de novo acquisition of protein-protein, co-immunoprecipitation, or other biomolecular interactions. While this is suitable for novel findings, it has limited applicability in high-throughput profiling, particularly if such interaction relationships are already documented. Networks can also be organized statistically, for instance using co-expression parameters in much the same way as heat maps are assembled. These can yield new insights suggested by co-regulation, but do not guarantee that a biological relationship exists between co-expression partners. A third approach to network assembly is ab initio modeling, reverse engineering interactions via expression dynamics. This requires extensive expression profiling over numerous perturbations, either time points or drug doses, or an additional perturbation, that may be beyond the scope of current high-throughput approaches to yield sufficient data for reliable modeling. A fourth approach, and likely the easiest and most common at this point, is to use current biological knowledge to assemble differentially expressed data into known interactions. This can be carried out from pathway analysis data, literature-generated interactions, or via existing interaction databases such as String, Reactome, or HPRD, the Human Protein Reference Database. As mentioned earlier, comprehensive pathway analysis tools are excellent for extracting network interactions, but are not ideal for assembling or visualizing large networks, nor are they intended for interrogation of network structural properties. Network-specific tools combining visualization with complex network analysis are thus particularly advantageous in this regard. An ideal example of this is Cytoscape. Cytoscape is an open-source software platform for visualizing molecular interaction networks and biological pathways, and for integrating networks with annotations, gene expression profiles, and other state data. It was originally designed for biological research, but now has much broader applicability. It comes with a basic set of features to upload, visualize, and analyze your data, but since its introduction in 2003, the many apps that have been developed as plugins really add to its power. Most apps are freely available, and some that were originally plugins are now fully integrated components. We will look at two of these apps here to get a sense of how to use Cytoscape to examine the underlying structure of a network and how you can interpret functional aspects of networks from within Cytoscape. We will upload existing proteomic datasets and examine them using these two tools in the hands-on portion of the bootcamp. First though, here's a snapshot of the Cytoscape 3.5 desktop. There are various options for data input, and we'll upload our own data, but Cytoscape is now interoperable with other bioinformatic tools, allowing you to directly connect with external databases if desired. Node and edge annotations can be added and applied using specific layout tools and network characteristics are readily accessible in table format below the network image. Certain apps will generate new windows of analysis graphs, tables, or image output. Cytoscape is extremely versatile for mapping and layouts, color coding, export of images, and the application of filtering or other network adjustments on the basis of corresponding expression data. Now we'll take a look at a couple of the more useful Cytoscape apps. First designed as a Cytoscape plugin, Network Analyzer performs analysis of biological networks and calculates network topology parameters, including the diameter of a network, the average number of neighbors, and the number of connected pairs of nodes. It also computes the distributions of such features as node degree, as shown here, average clustering coefficients, topological coefficients, and shortest path lengths. Results are available primarily as graphs, but can be exported or saved as images or text files. A number of Cytoscape apps perform similar or more specific tasks, but Network Analyzer is probably the most complete for complex network analysis and is now integrated in every instance of Cytoscape. As you might guess, this particular network is too large to visualize in the pathway analysis application from which it was originally derived, and at first glance appears to be nothing more than a large furball. Shown beside the network is its degree distribution, which nicely demonstrates the scale-free approximation mentioned earlier in the description of network architecture for this particular network. Remaining with the same network in Cytoscape, we are now looking at output generated using the Cytoscape gene ontology tool known as Bingo for bioinformatic network gene ontology. There are also a number of gene ontology tools available as apps for Cytoscape, but this is one of their most popular downloads. Bingo generates gene ontology networks from the original interaction network. These secondary networks represent gene ontology terms enriched on the basis of user-specified search parameters, including selected organism, a gene ontology category of interest, again this could be biological process, molecular function, or cellular component, and the statistical test applied. And it plots a color gradient from yellow to dark orange to indicate gene ontology category nodes that reach a specified statistical threshold. And this color gradient default covers 5 orders of magnitude. These particular gene ontology networks are arranged using a hierarchical layout to present less specific parent terms at the bottoms of the images, with specificity increasing as you move higher up these acyclic directed networks, which would be considered downstream within gene ontology. Due to the interrelationships between nodes, often an entire branch will be overrepresented. In part because of this, the Bingo app creators suggest that the most intensely colored nodes farthest down the hierarchy, or up in these particular images, are probably the most relevant for understanding the biological enrichment within the original network. The data in these gene ontology networks can also be exported as tab-delimited spreadsheets to be visualized as histograms or tables, rather than as networks of networks. We hope that this introductory presentation has given you sufficient background to feel comfortable with the classroom portion of this bootcamp, where we'll have you use some elements of these tools to interrogate actual proteomic datasets, guiding hypothesis generation for experimental follow-up. See you at the bootcamp.